Hello everyone, how are you? Yes, today I am here to discuss an important and interesting topic with respect to medical entrance examination. The topic today which I am going to discuss is a part of the light reaction of photosynthesis. So let's begin with the topic. Do you know where photosynthesis takes place? Yes, it takes place in the green aerial parts of the plant, but the main organ of photosynthesis is the leaf. Why? Because leaf has an expanded surface area and it has the ability to trap maximum number of photons. Yes, we can take up with a diagram that is like this is the apical meristem, this part. This is a leaf. You can see that it has an expanded surface area and therefore it can absorb maximum number of photons from the solar radiation. Now, leaf also has a fine internal anatomy for photosynthesis. If we look into the section of the leaf, we find that it consists of upper epidermis. It is a dicot leaf, so it has the upper epidermis, the lower epidermis. It is also present, where the number of stomata is highly concentrated in the lower epidermis. In case of monocot leaf, we know that the upper epidermis and lower epidermis have equal number of stomata, that is isostomatic, but in case of dicot leaf, they are hypostomatic. So this represents the upper and lower epidermis, and here you have the vascular bundle, and these are the elongated cells, parenchyma cells, which are tightly packed or closely arranged. So these cells, they are representing the palisade parenchyma. Here the vascular bundle, the pith, the xylem part is present outside, the phloem is present inside. And below this, there are some parenchyma cells with rounded eminence and having profuse intercellular spaces. These cells are regarded as spongy parenchyma. So you can see that there is presence of two types of parenchyma inside the leaf. This is the upper epidermis, which has a cuticle also. It sometimes bears stomata. And inside this particular structure, this is a vascular bundle, and here the phloem is present inside and xylem is situated outside. In between the cambium they are present. So this is the upper epidermis. This represents the lower epidermis. And these elongated cells, which are tightly packed, they are regarded as palisade parenchyma. While the rounded cells parenchyma cells which are with profuse intercellular spaces are known as spongy parenchyma. Now this palisade and spongy parenchyma together constitute, yes, can you tell me what it constitute? Together constitute the ground tissue of the leaf, that is known as mesophyll tissue. So this two together will constitute the mesophyll tissue. And mesophyll tissue is the tissue where photosynthesis takes place inside the leaf. Because these cells are enriched with chloroplast, and chloroplast have a definite pigment, a particular pigment which is known as chlorophyll, along with many accessory pigments which are associated with it. So let us take one of the cell to have a proper understanding of it. Suppose we are just taking one of the cell of this mesophyll tissue. So this is a plant cell. I am not going to make all the organelles because all the organelles are not required for this purpose. We need to have that particular organelle which is mainly concerned with this process, and that is, this is a vacuole which is present in the plant cell, which is very big, nucleus will be present, but the most important organ, the most important organelle associated within the cell of the plant which is concerned with photosynthesis, it is known as plastid. This is a double membrane bound structure. This is a double membrane bound structure. You have studied in the cell chapter that plastid is an organelle which is found to be present in the plant cell, and it is of mainly two types. This is plastid, and it can be of two types. One is colored plastid and another one is non-colored. So this colored plastid involves again two types or categories. One is the chloroplast and another one is chromoplast. While the non-colored plastid, which is found to be present in the root, they are the leucoplast. Now this leucoplast can be of three types. When it stores starch, it is known as amyloplast. When it stores protein, it is known as alluroplast. When it stores fat or lipid, it is known as elioplast. So amyloplast, it is for starch. Alluroplast, it is for protein. Because they are present in the root, they are mainly concerned with storage. And the third one is elioplast, it stores fat. But our main concern for photosynthesis is chloroplast. Yes, we are going to focus our concentration on chloroplast. Now the chloroplast is a double membrane bound structure which contain the pigment chlorophyll and other accessory pigments. So first let us take up with the diagram of the chloroplast, and then from there we proceed further. So it is a double membrane bound organelle. It consists of some sac-like structures. The sac-like structures which are present within the chloroplast They are arranged in this manner in form of a piles, or sometimes they are single also, but mostly they are arranged in form of pile, and the sac-like structures appear like this, which are being connected by membranous network. Like this. Now here inside the structure of chloroplast, we see that it comprises of membrane bound structures. Now these sac-like structures, which are referred to as thylakoids. They are also known as grana lamellae, now this stack of thylakoids together constitute what is known as granum. This is known as granum, and number of granum, you can see that this is one granum, this is another granum, all these granum, this is singular, all these granum together constitute what is known as grana. So grana represents plural form, right? Now these granum, they are interconnected by a membranous tunnel like structure which is known as stroma lamellae. Which is also known as fret. Other than that, you know that inside the chloroplast there is double-stranded circular DNA which is present. Ribosome is also present, which is 70S type, and apart from that there is presence of some special type of lipid globules called plastoglobuli, and this dotted homogeneous part, it represents the matrix, which we can also say it is known as stroma. So this is the matrix or stroma. Matrix. So the part which I am forming over here is the matrix of the chloroplast. It is a site where dark reaction takes place, that over here, because we know that light reaction and dark reaction, these are the two events of photosynthesis. So the dark reaction, which is a light independent reaction, takes place over here. So site of dark reaction. While this grana, it is the site of light reaction. This is double-stranded circular DNA, and this represents the plastidoribosome. Now here we are going to discuss that inside this single thylakoid, before discovery of the electron microscope, it was found that there are certain small sac-like structures which are present within the thylakoid. They are referred to as quantosome. That means if we take a single thylakoid or a sac-like structure, this is a single sac-like structure, or this is a single thylakoid which also consists of two membrane. Now within this thylakoid, initially it was believed that there are small sac-like structures, and these small sac-like structures, they were initially referred to as quantosome. Now if we look into, this is also known as the photosynthetic unit, and if we look into a single quantosome, at the center there is presence of a single molecule of chlorophyll. Single molecule of chlorophyll at the center, and this center is known as the reaction center. Yes, the photochemical reaction center I referred to. So this is the main pigment. It is surrounded by some accessory pigments. Yes, I'm giving this dot structure. It is representing the accessory pigment, and there are about 100 such accessory pigments which are known as light harvesting complex. Individually these pigments are known as light harvesting pigments. So they are known as the light harvesting complex, or you can also write it as LHC. They are also known as accessory pigment, or simply they are also called antenna pigment. Now these antenna pigments, along with the main pigment, they are associated with some proteins. So the main pigment is present at the photocenter or photochemical reaction center. It is surrounded by light harvesting complex made up of light harvesting pigments, and these light harvesting pigments are associated with proteins. So the entire thing constitute what is known as photosystem or light harvesting system. They are known as photosystem. So these are the proteins. And there are two types of photosystem. Now the entire thing, it is known as the photosystem, and there are two types of photosystem. Yes, one is photosystem 1, we can refer to as PS1, and another is photosystem 2 or PS2. Now this photosystem 1 is concerned with absorption of 700 nanometer of light. That means at 700 nanometer, this photosystem, the chlorophyll A which is present, the single molecule of chlorophyll A which is present at the reaction center and serving as the main pigment, gets activated. So on the basis of activation, on what type of wavelength of light, we have classified the entire light harvesting system or the photosystem into two categories. One is photosystem 1, which is concerned with absorption of 700 nanometer of light, while another is photosystem 2, where the main pigment is concerned with absorption of 680 nanometer of light. That means at 680 nanometer it functions very well for the purpose of photosynthesis. Now after this, with the advent of electron microscope, we find that the concept of this quantosome was miscellaneous, and the entire pigments and the carrier complex which are responsible for transport of electron, they are found to be located on the membrane of the thylakoid. So I am just going to take up that part over here. Now here we can take up with the membrane of the thylakoid, where I will be showing you that the different pigments and the carrier complex, they are arranged in a definite pattern. So let me take up the diagram of that. See, I am going to take up the pictorial view of here. I am forming the thylakoid, which is a double membrane bound structure. a single thylakoid membrane. It consists of two parts, that is the outer membrane and the inner membrane. Right? It would be convenient that if I rub this part, now all these carriers and the pigment system, which we have already discussed, they are located on the membrane of thylakoid. And if we just think about the position of the photosystem, initially we know the previous concept, according to the previous concept, that they are present inside the thylakoid. That means the pigment systems are concentrated within some small vesicles. Initially it was proposed that there were some small vesicles which were referred to as quantosomes. Now these quantosomes, they contain certain pigments, and these pigments are having the main one, which is located at the center, which is known as the photocenter, and the accessory one which is present outside it. But later on, with the advent of electron microscope, it was found that all these, that is pigment system, that is photosystem, and the electron carrier, they are present on the membrane of thylakoid. Now inside the chloroplast, we know that the pigment system, they are arranged along with the carriers, and they are arranged according to their redox potential. In our previous diagram which we have discussed, I have shown you that the thylakoid membrane, it consists of two layers, the outer and the inner layer, and this part can be represented like this. They are also connected to one another. So if I just make it like in this way, we find that there is connection between the thylakoid membrane, or we can say that one granum is connected with another granum to constitute what is known as the network or granum. I have already discussed this diagram before, but the thing which I need to discuss over here is that, see, this is a chloroplast. So this represents the thylakoid, and this part, where the topmost part of the first thylakoid, suppose this I am marking it as first thylakoid, this is the second, and this one is the third. This part which is free, and there is no other thylakoid which is present over here, this part, it is known as unappressed part. You can write it in this way, unappressed. The unappressed region, pigment system 1 is present, but pigment system 2 is absent. What are the things which are present in the unappressed region? What do you mean by unappressed region? That means the region of the thylakoid which is not being suppressed, or there is no other thylakoid which is present over it. So it consists of following things, that is photosystem 1 is present. Along with that, there is presence of cytochrome, BF complex. This particular complex, it is also present. And there is another complex which is responsible for ATP synthesis, that is CF1 complex. Yes, this is also known as ATP synthetase. We can also call it as ATP synthase because the main function of this complex is to synthesize ATP. Along with that, I can write it as ATP synthetase. Along with that, there is presence of FNR. What is that? That is ferredoxin NADP reductase. You can simply call it as NADP reductase. It is also present on the unappressed region. So ferredoxin NADP reductase. it is present. But what about the appressed region? Yes, this part, it can be represented as appressed region, because the third thylakoid, if I just show you the second one, the topmost part of the membrane of the second thylakoid is being appressed by the third one. So this is known as appressed part. And appressed part consists of photosystem 2. Yes, it is a pigment system 2. So what are the things which are present associated with the appressed part? That is known as the photosystem 2, and along with that there is presence of cytochrome BF complex. Yes, this complex is also associated with the photosystem 2, and cytochrome BF complex is present in the appressed region. But what about this part? Yes, it is connecting the two granum. It is known as fret. It is also known as stroma lamellae. And over here, photosystem 1, PS1 is present. But what is absent is that NADP reductase, or you can simply call it as FNR, it is absent. So what is present over in this stroma lamellae consists of PS1, it is present, PS2 is absent, PS2 is not present over here, PS2 and FNR, yes, FNR, they are absent. So this is the basic thing we need to know, that what are the pigments, how they are arranged. And another thing is that on the basis of the chromatography, it is known that the color of chlorophyll A is basically, it is bluish green in color, while the color of chlorophyll B is yellowish green in color. Carotenoids consist of carotene and xanthophyll. Xanthophyll is yellow in color and carotene, they are orange in color. So basically photosystems are arranged in this manner. On the unappressed region, photosystem 1 is present, cytochrome BF complex is present, along with that ATP synthase and FNR, they are present. In the appressed part, photosystem 2, cytochrome BF complex, it is present, and along with that there is also presence of OEC. That means it is associated, it is present on the inner side of the thylakoid, associated with photosystem 2, which is also known as oxygen evolution complex. It is present. And in the membrane of stroma lamellae, we can call it as the stroma lamellae or fret. Only PS1 is present. PS2 and FNR, that is NADP reductase, it is absent. After this, let us take a single thylakoid and try to understand how the process operates and the light reaction of photosynthesis takes place with respect to chemiosmotic hypothesis. Now we are going to discuss chemiosmotic hypothesis. Last discussion was that we have going to take a single thylakoid, and I am going to highlight it in a big version. So let us take up this diagram now. So we are going to discuss chemiosmotic hypothesis. This hypothesis was proposed by Peter Michel, for which he got Nobel Prize, and this explains nice that how ATP formation takes place in presence of light. So I am going to form over here a single thylakoid, and we know that from the modern or current view, all the pigment system, they are present on the membrane of thylakoid. So see, this is the outer membrane of thylakoid, and this is going to be the inner membrane of thylakoid. So this is the inner membrane of thylakoid. And just now I discussed the position of photosystem 1 and position of photosystem 2. So what we have seen that photosystem 2 is present slightly embedded in the thylakoid membrane, while photosystem 1 is present at the upper region, that is unappressed region. So I can put the photosystem 2 over here. This is the photosystem 2. So I am writing as PS2, and this represents the photosystem 1. You can see that the position of this photosystem 1 is slightly above that of the photosystem 2. I am rubbing this part, that means this write-up of Chemiosmotic Hypothesis, so that I get some space over here. And at this region there is presence of a carrier complex which are present across the membrane in form of transmembrane carrier complex. This is a carrier complex which is present on the membrane. They are arranged according to their redox potential. This is known as cytochrome BF complex. This is known as cytochrome BF complex. And as soon as this photosystem 2, we know that when the light energy strikes the photosystem, they gets excited. So when this photosystem 2 gets excited with the help of light wavelength of 680 nanometer, that is 680 nanometer of light, when falls on this thylakoid, it elicits, or you can say that it activates the photosystem 2. And being activated, the chlorophyll A, suppose there are accessory pigments and the main pigments are also associated. The main pigment, that is the chlorophyll A, which is the ionic configuration. If we just consider about the electronic configuration of the chlorophyll A, we see that this is the ground state. As soon as it acquires some amount of energy, the electron is raised over here, and this state represents the second singlet state. I am writing SS, it stands for singlet state. So from the second singlet state, some amount of energy, because this state is highly energized, and from this particular state some amount of energy is released in form of heat. So we can write over here that minus delta G, that this amount of energy is released in form of heat, and it will come again in another state, into another configuration energy state of electron, that is known as first singlet state. From the first singlet state, it can come either to the ground state by release of light energy in form of light wavelength, which is having greater amplitude than that of the incident wavelength of light. That is known as fluorescence. And if it reaches another state, sometimes it can reach another state also, if it is that another state can be reached by the main pigment as well as the accessory pigment, that is known as the triplet state or third singlet state. This is known as the triplet state. Now in the triplet state, we know that inside an orbital, the electrons are present in half, plus half and minus half in this orientation. They show plus half and minus half orientation of arrangement. Now they rotate in that way, that is in this condition they are present. In the opposite manner, this is known as spin quantum number. They are always in the opposite manner, the rotation of the electron. But in this state, what happens, it tends to rotate in the same direction, and therefore they become unstable. Being unstable, the electron leaves the chlorophyll and goes out. So what happens, usually in an orbital, the electron pairs, they move in an opposite direction, and in triplet state they tend to move in the same direction. They becomes unstable, as a result of which the electron goes out, and this is known as Pauli's exclusion principle, due to which the chlorophyll, that is a protochlorophyll, which is inactive form of chlorophyll, they gets activated and they becomes CH plus. If I just extrude 2 electron, I can write CH double plus. So this process is known as photoionization, because in presence of light, chlorophyll molecule, they are getting ionized. So I can write it as photoionization. So this is photoionization, and this is when the light energy is emitted with larger wavelength than incident ray of light, when the electron directly from the first singlet state comes down to the ground state. Or in some accessory pigment, we can say the antenna pigments or light harvesting pigments, from the triplet state also, due to the lack of better reaction partner, they cannot encounter better reaction partner, so they can come down over here in the ground state. They will emit radiation of light or energy in form of greater light emission of wavelength. They will emit energy in this case, if they comes down from the triplet to ground state, they will emit energy in form of light which is having greater amplitude than that of fluorescence as well as incident light energy. So from the triplet state, sometimes in the accessory pigment or light harvesting pigment, it is also found that the ray, that means the energy is radiated with larger wavelength. This particular wavelength is having larger amplitude or greater amplitude than that of the fluorescence and incident wavelength of light. This wavelength is known as phosphorescence. So fluorescence and phosphorescence together, we can take it as photoluminescence. Or we can also call it as bioluminescence. So this is the way we can say that the electrons, they are extruded out and the chlorophyll becomes excited. So this is light activation of chlorophyll, where the protochlorophyll, which is inactive form, which is present in inactive form, in presence of light, they loses electron and they becomes active form of chlorophyll. Now this particular active form of chlorophyll is ready to take part in photosynthesis. So this is the active form of chlorophyll. Now after this discussion, we can come over here in this chemiosmotic hypothesis, how this is operating, where we can see all the photosystem and the carrier complex, they are arranged on the membrane of the thylakoid. So I would like to rub this part. Now when the light radiation of 680 nanometer comes and strike this photosystem 2, along with that there is another light radiation of 700 nanometer, because you know that at this 2 wavelength, the 2 photosystem gets best activated. This photosystem 2, the chlorophyll A of the photosystem 2 will get activated initially. This is very important to note over here, that initially all the carrier complex which are present on the membrane, they are arranged according to their redox potential. That means the redox potential of photosystem 2 is greater till light have reached. When the light have striken the photosystem 2, its redox potential decreases, ionization potential increases. I repeat this again over here. Listen to this again over here. I said whenever the redox potential is low, the ionization potential, it is going to be less. Suppose we are taking that is the concept of an atom. When the atom is energized with respect to some other energy from the photons or some other sources of energy, the energy contained or energy potential of the atom will increase, and this energy potential is inversely proportional to the redox potential. So I can write it over here, inversely proportional to redox potential. And and redox potential is directly proportional to ionization potential. So I can write that redox potential, it is directly proportional to ionization potential. So energy potential is inversely proportional to ionization potential. So what is ionization potential? It is the tendency of an atom to lose the most loosely bound electron, right? So over here, initially the redox potential of photosystem 2 is more, but when it is stricken by the light wavelength, the redox potential gradually decreases with respect to what? The first electron carrier, which is going to accept the electron, because the carrier which is having greater redox potential, it can accept the electron from the another partner, reaction partner, which is having lesser redox potential. I repeat this part again. In a reaction, when the substance which is having lesser redox potential is going to lose electron, and another one which is having greater redox potential is going to accept the electron. So now when this photosystem 2 gets excited, the electron will be extruded from this photosystem 2 and it will be first accepted by pheophytene. So as soon as the photosystem 2 gets activated, its redox potential decreases, the redox potential of pheophytene increases, and a pair of electron is extruded from the photosystem 2 and is accepted by pheophytene. Then it will go to a mobile carrier that is known as plastoquinone. We know that the substance which is having greater redox potential has the better tendency to accept electron and retain them. So therefore this pheophytene is having now at this present the better retention power of the electron. From the pheophytene, the electron will be moving towards plastoquinone, and as soon as the plastoquinone, it is a mobile carrier, it will take up 2 electron, it will at the same time it will extrude 2 protons from the stroma. So from the plastoquinone, because this part it is going to take place in the stroma, stroma, this is also known as the matrix. Right? Now this plastoquinone is going to accept 2 electrons, and at the same time it will extrude, it will take up 2 protons from the stroma and it will become plastohydroquinone, PQH2. Now this plastohydroquinone, it has a property of retaining 1 electron with it, and there are 2 plastoquinone molecules, it will retain 1 electron within the system and another electron will be given to cytochrome BF complex. That means 1 electron will be moving down to cytochrome BF complex because it is a one system carrier. Plastoquinone, pheophytene, they are 2 system carrier because they can accept 2 electrons, and plastoquinone being a mobile carrier, when it accepts 2 electron, it will be having the potential of picking up 2 proton from the matrix. Right? Now this particular carrier, that is cytochrome BF complex, which is present, it is not a mobile carrier, it has the ability to accept only 1 electron at a time and it do not have the ability to accept proton. So what is going to happen with the proton? Yes, this particular that is plastoquinone is going to get converted into semiquinone. I can write it like this, that is Q minus, this is known as semiquinone. This is going to wait for the again another electron, and these 2 protons which it has taken, it will be pumped down into the lumen of the thylacid. So 2 protons are pumped per electron transported to cytochrome BF complex. Listen to here again, that means plastoquinone, it is receiving 2 electrons. At the same time it has the ability to pick up 2 protons from the matrix. As soon as it picks up 2 protons, it will get converted into PQH2, which is known as plastohydroquinone, and again it will be releasing 1 electron to cytochrome BF complex. At the same time the protons which it has taken up, it will be pumped down into the lumen of the thylacid, and 1 of the electron will be retained within the system, and this will remain as the semiquinone. So this cycle goes on, which is represented as Q cycle. So this is called Q cycle. Due to this, per electron transported from the PQ, that is plastoquinone, to cytochrome BF complex, 2 protons are transported across the membrane into the lumen of the thylakoid. Yes, it represents a lumen, and this is the membrane of the thylakoid. And after that, from the cytochrome BF complex, it is going to move down to plastocyanin because it is having greater redox potential, and from the plastocyanin it will come down to the photosystem 1. But what about the photosystem 1? Photosystem 1, to receive the electron, it has to have some vacant spaces with respect to electrons. As soon as the photosystem 1 receives 700 nanometer of light, it will again, similar to that, like that of the PS2, that is photosystem 2, it is going to lose electron. The electron is going to come out like the same way, and the first acceptor of the electron over here for photosystem 1 is FeS, iron sulfur cluster. So I can write it over here that this is FeS. It will be accepting the 2 electron at the same time. Now from the iron sulfur cluster, the electron will be moving down to another complex which is loosely associated, yes, which is loosely associated on the outer side of the thylakoid membrane. This is very important with respect to examination point of view, that this particular protein, which is known as ferredoxin, I can write it like FD. There are 2 molecules of ferredoxin. These 2 molecules of ferredoxin are associated on the outer side of the thylakoid membrane, associated with the PS1. Outer side of the thylakoid membrane, loosely bound on the outer side of the thylakoid membrane, associated with PS1. And to this ferredoxin, another enzyme system which is associated is known as FNR. Yes, you can also call it as NADP reductase. This is NADP reductase, and this particular enzyme is a multi-enzyme complex within which another compound, or you can say coenzyme, that is FAD molecule is present, FAD plus is present. The FAD plus have the ability to accept 2 electron and 2 protons. So FAD, that is flavin adenine dinucleotide, it has the ability to accept 2 electron and 2 protons, and there will be formation of FADH2, and ultimately from the entire complex, that is NADP reductase, which is having FAD as a part of its complex, will convert NADP plus to NADPH. NADP plus to NADPH conversion will take place. Remember one thing, NADP plus has the ability to accept, NADP plus has the ability to accept 2 electrons, but it can accept only 1 proton. It can accept only 1 proton. It can accept 2 electrons but only 1 proton, not like that of the FAD plus. FAD plus can accept 2 electrons and 2 proton at the same time, but when it will come to the NADP plus, it will be accepting 2 electron and it can accept only 1 proton. So what will happen to another proton? Yes, you can see that FADH2, that is this, what will happen to another proton? It will remain as the site like H plus ion. So the correct version is not NADPH2, the correct version is NADPH plus H plus. So there will be formation of NADPH plus H plus in this way, and per electron pumped from the plastoquinone to cytochrome BF complex, there is pumping of 2 protons into the lumen. Now can you tell me that to allow 1 electron flow from PS2 to PS1, how many photons are involved? There are involvement of 2 photons. That means PS2 and PS1 has to be activated simultaneously. So 1 photon, say this is one particular photon, this is another photon which is going to activate PS1. This is another photon of say 700 nanometer of light. This is 700 nanometer. And this is the first photon which is going to activate PS2. This is 680 nanometer. Already I have written it over here down. So there is requirement of how many photons? There is requirement of 2 photons, and as soon as this 2 photons is required for PS2 and PS1, there can be movement of 1 electron, because as soon as the electron comes out of this photosystem 1, there will be a gap for 2 electrons in the photosystem 1, and this gap will be filled up, or this electron deficit will be filled up by the electrons coming from its carrier, that is cytochrome BF, plastocyanin. Yes, it is another mobile carrier. So there are 2 mobile carrier. One is the plastoquinone, it is a mobile carrier. Plastocyanin, it is also another mobile carrier. Cytochrome BF complex is not a mobile carrier, it is present throughout the membrane, and this is blue in color because it contains copper. Plastocyanin contains copper, it is blue in color. So you can call it as blue carrier, and this cytochrome B6F, it contains iron, so it is called red carrier. So this is a red carrier. So in this way we can see that photosystem 1, the electron deficit which has been developed in the photosystem 1 is filled up by the electrons coming from the photosystem 2. But what about photosystem 2? The electron deficit has already developed in the photosystem 2, and that will be filled up by water. Now on the inner side of the inner membrane of the thylakoid, there is presence of another protein. We can say there is presence of another multi-enzyme complex which is known as OEC, that is oxygen evolution complex, which is also known as water oxidase. And with this there is involvement of certain elements like manganese, chloride, and yes, manganese chloride is present with this particular complex, and calcium is also present. So we can say that there is involvement of 3 elements. One is manganese, chloride, and calcium, which is associated with oxygen evolution complex. And if we talk about the position of the oxygen evolution complex, it is present on the inner side of the inner thylakoid membrane associated with the PS2, which is associated with oxygen evolution complex. And if we talk about the position of the oxygen evolution complex, it is present on the inner side of the inner thylakoid membrane associated with the PS2. I repeat, oxygen evolution complex or water oxidase, which is having manganese, Mn4+, Cl- and calcium present on the inner side of the inner thylakoid membrane associated with the PS2. This is going to break the 2 water molecules in this way, and there will be formation of 4 electrons, there will be formation of 4 protons, and 1 oxygen molecule that is going to get released. So 2 molecules of water can produce 1 molecule of oxygen. So how many photons will be released? How many photons will be required in this process? 2 photons are required for transport of 1 electron, and for transport of 4 electron, so 2 photons which are 2 photons are required for transport of 1 electron. So I can write 1 electron is requiring 2 photons for its transport from PS2 to PS1. So if it is a matter of 4 electron, so we can write 4 into 2, that is 8 photons will be required for transport of all these 4 electrons. So all these 4 electrons will be accepted by this one, that is OEC, and one by one it will reach the photosystem 2. In this way the electron deficit of the photosystem 2 is being filled up. Now how many H+ ion is present? Yes, 4 H+ ion is present over here. At the same time, if 4 electrons they flow, per electron 2 protons are pumped into the lumen of thylakoid. Yes, per electron pumped from the plastoquinone, plastohydroquinone or plastoquinone to cytochrome BF complex, 2 protons are pumped into the lumen of the thylakoid. For pumping 4 electrons, how many protons will be pumped? That is 2 into 4, that is 8 protons will be pumped. So over here 8 protons will be pumped across the membrane by plastoquinone or plastohydroquinone into the lumen when 1 electron is passing from it to the cytochrome BF complex. Now already how many protons are present due to photolysis? that is 4. So 4 plus 8, how much? 12. 12 protons are present now inside the lumen of the thylakoid. It is going to decrease the pH, because we know that with more hydrogen ion accumulated over here, the pH is going to be low. So it is going to create a low pH, and at the same time it is going to elevate the positive charge as compared to the stroma, because you know that stroma is having less H plus ion, because already H plus ion is pumped into the lumen of the thylakoid, and here H plus ion concentration is high. So in the NEET examination, often this question has been asked that where during the operation of this process H plus ion concentration is more. Yes, it is the lumen of the thylakoid where the proton concentration is high. Proton concentration is high over here. That means H plus ion concentration is high over here inside the lumen. So 12 H plus ion is formed, and they are going to generate electrochemical gradient. What do you mean by electrochemical gradient? Electrical gradient, that is more positive charge over here as compared to the stroma, and pH gradient. Chemical gradient means pH gradient. Electro means charge gradient. Charge is positive inside the lumen, charge is comparatively negative outside, and chemical gradient is pH gradient. pH is lesser because of more H plus ion inside the lumen, and pH is more outside. So this electrochemical gradient will allow the movement of the proton outside. That means there will be generation of an entropy which will allow the movement of the H plus ion outside from the lumen into the stroma, and that is going to take place by an enzyme complex which is known as F0 CF0 CF1 complex. This enzyme complex is known as ATP synthetase. This is the main function of this enzyme, is to synthesize ATP. The CF0 complex, C stands for chloroplast. The CF0 complex is a channel. It is a channel which is present embedded into the membrane of the thylakoid. It is a transmembrane channel, and when proton will be moving down through it, it is going to get active. And this part, which is the CF1 part, it has certain receptors. The receptors are present in this direction, in this conformation, which is not exposed. Now as soon as the protons they are going to move down through this particular complex, it is going to get activated, and the receptors are going to get exposed like this. So what it is going to do, how many protons are going to flow? Yes, 4 protons at the same time. At a particular time 4 protons are going to move down, and as a result this particular channel is going to get active. At the same time the receptors are going to get exposed. These are the receptors for which thing? They are the receptors for ADP, because they are going to decrease the activation energy. They are in enzymes, and this is inorganic phosphate. So ADP and inorganic phosphate, they will combine together. That will result in formation of ATP. So we can see that using the light energy there is formation of ATP. So we can also call it as photophosphorylation. Though light is always over here, it is mandatory, but it may be a condition which was performed by Dragon Drops, where he purposefully maintained without light low pH inside the lumen of the thylakoid, and found that ATP formation is also possible. So over here we can see that 4 protons are transported across the membrane, and there is formation of 1 ATP. So for 4 protons 1 ATP is formed. So how many ATP will be formed for fixation of 1 carbon? Because you can see how many protons are present, 12. So if 4 of them goes down, so 3 ATP will be formed, because 12 H plus can be divided by 4, that will come as 3. So there will be formation of 3 ATP. And just now I discussed NADP plus. 1 NADP plus is going to receive 2 electrons, only 1 proton, and 1 proton is remaining as proton gradient. So for formation, when one water molecule is broken down or photolysis takes place, there is release of 4 electrons. That means there is formation of, for 2 electrons there is formation of 1 NADPH. For 4 electrons there will be formation of 2 NADPH. and 2 H plus ion. So for 1 NADP plus to 1 NADPH requires 2 electron and 1 H plus ion. So over here there is, when 2 water molecules are splitted, there is formation of 4 electrons. So 4 electrons is going to produce 2 molecules of NADPH. So in this way we can calculate this particular thing. You can see that it is taking place in a Z-shaped manner. This is known as Z scheme. So if we see that the Z scheme, everything that is 2 water molecule is releasing 1 oxygen molecule, and for that how many photons are required? 8 photons. So 8 photons is going to release 1 molecule of oxygen. This is known as quantum requirement. This particular thing is known as quantum requirement. What is quantum requirement? That is, to release 1 molecule of oxygen, how many photons are required? It has been found to allow movement of 1 electron from photosystem 2 to photosystem 1, there is requirement of 2 photons. So to allow 4 electrons, there will be requirement of how many photons? 1 electron is going to move from here to here with 2 photons. 4 electron, 4s are 2, 8. So 8 is the quantum requirement. So if 8 photons are required to release 1 oxygen molecule, that means 1 oxygen molecule is released by 8 photons, then we can write that 1 photon is going to release how much amount of oxygen molecule? That is 1 by 8. So 1 by 8 amount of oxygen molecule will be released if there is only 1 photons. This is known as quantum yield. So there are 2 words, one is quantum yield, another is quantum requirement. Quantum requirement means 8 photons are required to release 1 molecule of oxygen, and if there is only 1 photon, there will be release of 1 by 8 amount of oxygen. So this concept is also known as Chemiosmotic Hypothesis. Osmosis means crossing the membrane. Here chemical substance is protons. Protons are crossing the membrane. As a result, there is formation of a gradient. Whenever there is formation of a gradient across the membrane, the protons will cross the membrane, and as a result there will be formation of ATP. So this justifies that the chemiosmotic hypothesis. Why this is called chemiosmotic? Chemical substance and moving across the membrane. So chemiosmotic, it is creating an electrochemical gradient, and it is proposed by Peter Michel. It is also known as proton motive force, because the protons, they are the motive force behind ATP formation. The protons are moving out from the lumen into the stroma. As a result, there is formation of ATP. So it is also called proton motive force. So this explains this theory well, and also we can have number of calculations from this particular thing, and concept can be well understood with respect to the light reaction of photosynthesis. We are going to continue with other problems which are associated with this concept, and which is quite very relevant with the concept of the photosynthesis and frequently asked questions.